Western by Tom Happy. So you can just take a look at this, just read it for 20 seconds. Hi everyone, so thanks for reading through the question. It's always good to just kind of quickly glance at it, because now we can actually extract some relevant information. So over here, we extract quite a bit of info about the question and we can get a lot of work done with it. So we know that the child has trisomies given to the question. She has 3 copies of one of her chromosomes. We also know that the parents are karyotypically normal, or that they do not have trisomy. We're also told that she has the same SNP and the same location on one of her chromosomes as her father. And that's really important because she has 3 chromosomes. So when you have one SNP on one chromosome, that tells you a lot about what the question is. So what's a really, really quick way and easy way to go through this question? Well, if you know what meiosis 1 and meiosis 2 are, you really can quickly get the effect of meiosis 1 and meiosis 2. So, to summarize this question, before we even get to it, a non-disjunction error in meiosis 1 will result in different SNPs in one of the gametes. Different SNPs. Whereas a non-disjunction error in meiosis 2 will result in the same SNPs. So if we see some possible genotypes over here for the child, we can have one allele be from the dad, then we can have another allele from the dad on the second chromosome, assuming that we get 2 from the dad, but this allele is different, as indicated by the circle here. Then we have 1 from mom. So that's assuming that the non-disjunction happened in the fall. Then, if we have an error in meiosis 2, we can assume that 2 of the dads are the same, mom is just there, and then the same thing goes with mom and dad. So, based on the information provided by this question, we can quickly infer that the answer cannot be meiosis 2 in the dad. And you might be asking, well, why? Well, remember, she said they have the same SNP on one chromosome as the father. One chromosome. What this is suggesting is that 2 alleles are the exact same on that chromosome. What that tells us is that they would also have the same SNP on the very same location, which defines it's against what the question told us. So we immediately know that meiosis 2 is incorrect. And all the other meiosis in the dad and the mom work out just perfectly. So by that logic, we can say that non-disjunction happened in either meiosis 1 in the dad, because it creates 2 different SNPs, meiosis 1 in the mom, because again it creates 2 different SNPs in the gametes, or meiosis 2 in the mom, because it creates 2 of the same and 1 of the dads, and they did not place a restriction on whether you can have 2 SNPs from the mom. And that's all you really need to do this question. Now tune in if you want more information on the background of how we got this table, or just how generally to approach meiosis questions overall. Hi everyone, welcome to part 3 of our video series on meiosis and meiosis problem solving for Bio 1000 and Western. All we're going to be doing in this video, or in this part of the video, is just going over meiosis, what generally happens, and how errors in non-disjunction for meiosis 1 and meiosis 2 can give us different results, and how those translate to the table that we previously used in the problem, number 1. So let's just go right into it. What does meiosis 1 do? In a regular cell, all meiosis 1 does is it splits this tetrad. So what happens is that we get one chromosome over here and 1 chromosome over here. What meiosis 2 does is it splits this chromosome based on sister chromatids. So one sister chromatid is going to go over here, and one sister chromatid is going to go over here. And so that leaves 4 gametes. Notice that these 2 gametes are also different from these 2 gametes. That's because these are maternal chromosomes or paternal chromosomes, and these are maternal and paternal chromosomes. They're basically different chromosomes because you undergo sexual recombination based on your parents' chromosomes within your own cells. So let's get right into non-disjunction meiosis 1 and what that means for our cells. Well, all non-disjunction actually means is that if fails to separate a chromosome or a chromatid in a certain part of meiosis. So if you look at non-disjunction meiosis 1, what's happening is that we fail to separate our chromosomes in meiosis 1. So what we're left with is one empty cell with nothing because there are no chromosomes going into that cell, and one cell with 2 chromosomes or additional chromosomes based on your number of chromosomes in the original cell over here. Notice that they're different chromosomes. However, different from meiosis 1, which separates those chromosomes, meiosis 2 separates the sister chromatids. So what's that going to do for us? Well, one chromatid is going to go over here to this gamete, but instead of having this one go also in here, which is what we would expect, I guess, what happens is that the chromatid from the other chromosome goes into this gamete. What that results is 2 gametes, again with additional chromatids in them, but also the fact that the chromatids are different from each other. If you can just visually see it, you kind of go over here, and these 2 chromosomes are different, the chromatids are different, and they're put into the same cell. So just remember, meiosis 1, non-disjunction meiosis 1, gives us difference. So let's go over to meiosis 2 and see why meiosis 2, or non-disjunction meiosis 2, gives us the same result. Look right over here, we can see regular cells, but all non-disjunction meiosis 2 does for us is that we fail to separate our sister chromatids. So what happens is, because these sister chromatids don't separate in meiosis 2, we end up with one cell having 2 chromatids and one cell having no chromatids. And by cells I mean gametes. And well, what does that mean for our gamete? Well, this still has 2 chromatids in it. It still has 2 pieces of, I guess, original chromosomal DNA. But the difference between meiosis 2 and non-disjunction meiosis 2 and non-disjunction meiosis 1 is that the gamete here, the 2 chromatids are homozygous, they're the same. Whereas the 2 chromatids here are heterozygous, so they're different. And that's all that actually gives us this table, because if we look over here, back on our table, meiosis 1, let's just say we undergo non-disjunction meiosis 1, for the dad, all that's going to do for us, as you see over here, is that one allele is going to be, I guess, D. The other allele is going to be different from D, and that's why we indicated using this circle. Whereas in error meiosis 2, pardon me for the cross, but error meiosis 2 will give us the same. And same thing for the mom. An error meiosis 1, or a non-disjunction error meiosis 1, is going to give us difference, and a non-disjunction error meiosis 2 is going to give us the same. So for general, just think about these areas of meiosis when you're going through these questions. Also realize that sometimes in meiosis, there can be recombination, especially in meiosis 1. Well, that's the only place it can happen. That could introduce little difficulties in the question. But as long as you have that in mind, recombination can happen sometimes, and you follow these steps, you should be good for any meiosis question. Thank you.